All right, it's a real pleasure to be here today. And for this unusual occasion, I'm going to start in an unusual way. And I will start with giving you a bit of a career perspective. And this is a sort of glamorous version of my CV. And the point I would like to make is, you can find on PubMed that I've worked with fantastic people and in fantastic institutions, but it's also been amazing fun, to live in various places, and it's a really rich experience. So I just want to highlight the fact that to me, mobility is a great perk of scientific careers. So my scientific career started in Strasbourg, east of France. I had the privilege of working with these two scientists, very different, as you might gather from these pictures, at very different stages of their careers. And so it was a really rich experience. I started working on transcription in a pluridisciplinary large institute that had core funding. This institute was started from scratch by Pierre Chambon. It was an amazing place. We were all equal. We all led our own work. We could think big. The sky was the limit, and no hierarchy, yet it's fair to say Pierre was the boss. So together with my colleagues of the fourth floor of IGBMCs, we were identifying transcription initiation factors, cloning their cDNAs, and that led us to reconstitute pretty much transcription initiation in the test tube. So it was really, being part of a very exciting era and, a fantastic start of a scientific journey. And then, of course, came the question, what am I going to do next? The career prospects in France were very dull at that time. And, in this big question, Pierre gave perhaps the one career advice he gave to me, but it's a really big advice that helped me in many ways through my entire career and still carries me today. Just focus on the science, the rest works out. And so in thinking about a postdoc, just think about the science. That became a very simple problem. I knew what I wanted to do. I wanted, I trained in biochemistry, hardcore biochemistry, test tube. It was great fun. You see here not one but three ice buckets, and this is not the bench, this is a trolley. There are two more ice buckets here on the bench. So, it was a great time. But what I wanted to do for my future career was to start with a physiological problem and dissect it down to its molecular components. So that's kind of a broad idea, but there were many options there and opportunities that I could explore. But with a taste for the unknown, I went to New York. Joined David Ron's lab, who was then in New York, because it just began to work on something that was totally esoteric, that he called the ER stress response, the response to stress in the endoplasmic reticulum. What on earth is that? But there were some indications that one element, the only one we knew at the time, was conserved from yeast to mammals. And so the idea was it could be important. And for me, I took a long time, I must say, to decide on my postdoc. But the idea was, well, I'm going to bring biochemistry, my brain and expertise and outworking ethos to a lab led by an endocrinologist. This should lead to something unusual. And so it worked out indeed really well, because in only a few years, my postdoc was 2.5 years, we went from a really esoteric observation to what is now known as the mammalian unfolded protein response, and it's now in textbooks. So, and this is a pathway that we have evolved to defend ourselves from the threat of abnormally folded proteins in the endoplasmic reticulum. So this was the beginning of a new line of thinking for me. And, I left New York to move back to France without much thinking, really, because I'd earned one of these really competitive lifetime positions, and I thought, I should give it a try. And the idea is, I could find time there to think big and see what happened. And so the question I left my postdoc with was, you know, if we have this sort of protein quality control pathways that protects ourselves from abnormally folded proteins, why on earth do we develop these diseases? Neurodegenerative diseases indeed start when we have these proteins that start to assemble and accumulate and form insoluble aggregates in various neurons. And turns out that aggregation is not an inevitable consequence of these proteins that form inclusions. They are indeed mostly soluble throughout our life, actually. And so the question I started my lab with was to actually study the mechanisms that underlie aggregation. How does aggregation start? And, you know, this is a problem at the origin of many diseases. And so I started my lab after my first child was born, my daughter Lucy, and started to go well, published a first paper, then a second child was on the way and a second paper. And at that point, I started to think, well, it's perhaps time to review my career choices. And I became an EMBO young investigator at that moment. And perhaps this was a very important turning point in my career because, you know, I didn't want to go, oh, it takes time to look for places and interview and all that. And I didn't want to spend a lot of time doing this, and my career just started, my work was going well. And so I thought, I'm going to go to that YIP meeting in 2005 in Heidelberg and meet top scientists from Europe and ask them about their institutes and is it worthwhile visiting. And this is when I met Sarah Teichmann, who mentioned that the LMB was actually advertising. And at that time, I was not traveling with my laptop. So I went to Emble and the library to read the advert, and it was something like that. The LMB was looking for groups, you know, working on understanding important biological processes at the molecular level to bring knowledge that are relevant to human health. I read this advert and I thought, this is me. That's for me. Okay. I emailed my husband. He was like, go for it, Cambridge. Yeah, okay. So I applied, and then I came to interview here in December 2005 with a 3-month-old. This whole visit was handled very professionally, and I was, you know, given the privacy my circumstances needed. And above all, I think I had the opportunity to focus on the science during these 2 days. And this was a time of my life. I must say, these 2 days were so amazing. I wasn't too keen about Cambridge, but I was keen about the LMB. So I moved in August 2006. I was here. And so I continued working on the beginning of, you know, what all the molecular bases that initiate aggregation, as well as developing a new line of research based on the following idea. I mentioned, you know, all our cells during all our life express potentially harmful proteins, and yet, you know, diseases associated with protein aggregation start late in life. So why is this? It's because we have these protein quality control systems. They work well. We have, you know, the unfolded protein responses one, we have chaperones, we have proteasome, autophagy, and so on, that handle these bad proteins that we produce at all times, but it works well for decades. But as we age or in diseases, somehow the system gets overwhelmed. And so it brought a very simple idea. Can we try and boost these natural defense mechanisms in order to correct from the deficits that underlie many diseases? And so this is a grand idea, right? But then reality kicks in, and how on earth are we going to do this? And so we set out to actually recapitulate, in a way, the molecular basis of these diverse diseases by either forcing cells to accumulate abnormally folded proteins or impairing protein quality control systems. And this leads to cell deaths, right? It's inevitably fatal. And what are we going to do with this? Well, actually, it provides a really robust model that we can use and ask, can we find ways to help cells surviving under these otherwise catastrophic conditions? And in this way, what I've done here is converting an organismal problem, neurodegeneration, in a cellular problem, and you know, converting this so-called intractable problem into a simple life-death problem. I must say, when I proposed this, some people were not entirely convinced, that's putting it mildly, but LMB supported this line of work, so you're going to hear what happened. So first, I want to say that I exploited this idea in various ways, conducting in some cases genetic screening yeast to find ways to help yeast cells surviving these catastrophes. And in this way, identified new components of protein quality control systems that turned out to be evolutionary conserved. And I want to pause here because it's still mind-blowing to me that, you know, I was in the neurobiology division and I was doing yeast work. And I think this really speaks for the place. You know, it's open-minded. One can do whatever one wishes so long as we focus on important problems. So I'm not going to talk about the yeast work today, but I'm going to talk about an approach that started in cells and has been implemented in numerous models of neurodegenerative diseases, and importantly last year was found efficacious in humans. So these are very exciting times. And this started when we identified a small molecule that protected mammalian cells from protein misfolding inserts in the endoplasmic reticulum. And we worked out how it worked. It turns out that the molecule prolongs a natural defense mechanism, which consists in phosphorylating EF to alpha. This is a stress signaling pathway, and we found that the compound was protective by inhibiting the phosphatase controlling the termination phase of this pathway. So it's a classical signaling pathway. Kinase is activated, phosphatase is terminated, and all we did here was to find small molecules that actually inhibit the phosphatase, targeting the non-catalytic subunit, and in this way simply prolong this naturally occurring defense mechanism, and as a result protect cells from protein misfolding insults by increasing protein quality control capacity. And you know, this was exciting, and we thought, you know, let's try and implement this in models of neurodegenerative diseases. And the first to do that in my lab was Indrajit Das, who tested a second molecule, cephin, in a model of a rare neurodegenerative disease, Charcot-Marie-Tooth 1B, and he observed remarkable benefit of the compound in all the phenotypes he examined. So from the behavioral to the molecular phenotypes, and this is because we target the mechanism at the origin of the disease, namely the capacity of cells to deal with the burden of misfolded proteins. Numerous labs have followed up, and the list is longer than this, on these molecules, reported the benefit of the compounds in a variety of models. And I'm a huge fan of the work that Brian Popko did in models of multiple sclerosis, because he went beyond what we had done in showing that the benefit of guanabans and cephin in models of multiple sclerosis is abrogated in mice knocked out for R15A. So it's a strong argument to say that these mechanisms, these compounds act by an on-target mechanism. And more recently, we developed molecules that target the related protein R15B. The reason is that R15A is not expressed in all cells and all conditions, and we want to expand on the approach, targeting a protein which is expressed in all cells and all conditions. This has perked interest of clinicians that have embarked on various clinical trials and more on their way, and it's been immensely rewarding for us to see last year the report of a phase 2 clinical trial in amyotrophic lateral sclerosis, the success of a clinical trial 10 years, months for months after the first paper showing the protective effects of guanabans against misfolded proteins. It's been, this is an amazing study. In this study, they have 18 patients with bulbar onset that simply, for which the disease simply did not progress. And ALS, unlike Parkinson's, Alzheimer's, and so on, which are slowly progressive diseases, ALS progresses in a fast and furious manner. And so halting the disease for 6 months is really encouraging, and we've been really excited to see that. So it was worthwhile to work on this. There's independent evidence coming from others, and particularly like this one, supporting this line of work. It's a gene expression study from the lab of Michael Hayden in a group of 4,000 Huntington's disease patients that has revealed that some have delayed onset, and in these he found an increased expression of an EF2 alpha kinase. And so this provides human genetic data supporting the benefit of increasing EF2 alpha phosphorylation to slow down neurodegeneration. So we have now converging human genetic data as well as experiment data in cells, mice, and human to support this idea. But this is not going to be my closing note. I want to close on this. This whole work started with curiosity-driven expedition aimed at identifying some novel biological processes. This inevitably brings drug discovery opportunities, but there's more fun to have because these expeditions then in turn bring more fundamental question. And one we've been really interested in to develop was to figure out what was the function of these non-catalytic subunits of the EF2 alpha phosphatases. All we knew when we started was that they bound PP1, but the catalytic subunit, but we found that they harbor a very important function, which is to recruit the substrate. And we are, a long-standing effort of the lab is now to elucidate the mechanism by which this occurs. We are dreaming of getting a structure at some point, but there's another challenge on our way. The substrate recognition module of our favorite protein is predicted to be disordered, and preliminary NMR experiments suggest that this is indeed the case. So why do we care? Well, there are hundreds of such holoenzymes for which we know next to nothing. So the idea is what we learn on R15 might be relevant to the others. So why LMB? I think there's something very important to me is that we can really choose whatever we choose to work on, we can do it in depth, and we can follow the data and do whatever it takes on whatever question we choose to pursue. This is huge. And I want to close it saying that we are very fortunate also in being able to attract amazing scientists from all over the world. And I'm grateful to my current lab members as well as the previous lab members. Thank you very much.
Thank you for your talk. I just wondering, so you are saying that phosphorylation of 2 alpha is beneficial in prolonging the stress response. Can you comment on E3 studies which showed
yes,
actually the opposite?
Yes. There is EF2 alpha phosphorylation is a hub in cellular fitness, and not surprisingly, it is considered to be a hot target. And so we are prolonging this pathway. There are opposite approaches consisting of inhibiting the kinases or inhibiting signaling downstream of this pathway. And I think it is obvious that there will be therapeutic indications for all of these approaches, but in different indications.
Thank you. That was a very nice talk. I was wondering, so one of the key diseases that people often study with protein aggregation is Alzheimer's. But then there's been some discussion whether the aggregates, that's a symptom or that's a cause of the disease. And also recently that some of the key papers were falsified originally. So do you think this approach or looking into targeting the aggregates could be translated to Alzheimer's as well? Because that's one of the...
So there's 2 levels of answer to this question. One is, Alzheimer's has 2 deposits, an extracellular one and an intracellular one. I don't think the approaches I'm talking about can affect, or at least there's no evidence for me to suggest that these approaches could affect the extracellular deposit, but the intracellular one. The question about aggregates may not be causative of the disease is actually one of the reasons I came to LMB to work on. Max Perutz was, one of the discoverer of the fact that Huntington aggregates. And there's this whole discussion about what is the toxic entity in these diseases. There's this whole idea that oligomers may be the toxic species, but ongoing work from the lab, I think I can talk about it because it's on bioRxiv, from Michel's lab and others, reveals that what was supposed to be oligomers are actually fibrils. So I think this question as what is the toxic entity is going to be resolved soon. And I think, resolving the aggregates and finding ways to help cells coping with that is going to have a general impact on a range of neurodegenerative diseases.